So far, we've learned how to analyze inheritance patterns for one trait. In this video, we're going to analyze inheritance patterns for two traits, such as the color and shape of peas, which can be green or yellow, and wrinkled or round. One important place to start is choosing your symbols to represent the traits wisely. If you're just doing a one-trait cross, you only need one letter. So if we're only focusing on the color of the piece, we need a dominant Y to represent the dominant color, yellow, and then the lowercase y will simply represent the other version of the trait, green. But if you're doing a two-trait cross, you need, you guessed it, two letters. So in this case, we're using the letter Y again to represent the color of the pea, but then we're going to use a different letter, R, to represent the shape of the pea, whether it's round or wrinkled. Here's an example of what a monohybrid cross versus a dihybrid cross would look like. In a monohybrid cross, we're only looking at one trait. And so a monohybrid cross is where both parents are heterozygous for that trait. In this case, these peas are heterozygous for their purple color. A dihybrid is when the parents are heterozygous for both traits. So in this case, both parents are heterozygous for their round shape and they're heterozygous for their yellow color. Not all two-trait crosses are dihybrid crosses, but we are going to focus on that in this video. Another trick to doing two-trait crosses is genotyping the gametes, and we're going to take this back to meiosis for a moment. Let's say that this is our parental cell, our diploid cell, so it's got homologous pairs. Now, in the case of our two traits, one pair of chromosomes has the genes for color. Another pair of chromosomes has the genes for shape. And since this parent is a dihybrid, they're carrying a dominant and a recessive gene for color, and they're carrying a dominant and a recessive gene for shape. Now, if this cell divides by meiosis to produce haploid gametes, there are a few different combinations. Remember that the gametes have to get one chromosome from each pair, but because of independent assortment, different combos can result. Here are those combos. The dominant gene for color could end up with the dominant gene for shape. Or the recessive gene for color could end up with the recessive gene for shape. The recessive gene for color could end up with the dominant gene for shape. And the dominant gene for color could end up with the recessive gene for shape. So if we were genotyping these gametes, here are the possible results. The dominant R could go with either the dominant Y or recessive Y, and vice versa. The recessive R could go with either the dominant or the recessive Y. This is actually the hardest part to solving a two-trait cross. If you get this right, the rest is pretty simple. Here's another example of how to genotype a parent. In this case, the parent is green and wrinkled, our two recessive traits. So, in each case, all of the chromosomes are carrying the recessive genes, which means in our gametes, there's only one possibility. This parent can only make a gamete with a recessive Y and a recessive R. That's pretty simple. All right, so now that we know the basics, let's take a look at an experiment that Gregor Mendel did with two traits. So he started off in his parental generation with a purebred round yellow pea plant, and he crossed it with a purebred wrinkled green plant. Now, the results here are pretty easy to predict. Since this parent can only make gametes with dominant genes, and this parent can only make gametes with recessive genes, all of the offspring were dihybrids. They were heterozygous for both round and yellow traits. So then Mendel crossed these plants with each other. And here's what he got for the F2 offspring. A really interesting pattern, 9, 3, 3, 1. How did he end up getting ratio like that in his offspring? Well, let's do a Punnett square and find out. So with all Punnett squares, we're going to start with the genotypes of our parents, our dihybrid parents, heterozygous for both traits. Then the second and key step is to figure out the gametes. Now in both cases, they can make 4 possible gametes. So this male plant could produce these 4 gametes, and this female plant could produce these 4 possible gametes. These become our rows and columns of a Punnett square. And then the rest is simple. We just have to cross our male gametes with our female gametes. And here are the results we would get. As you can see, there's 16 possibilities. And if we start counting up for phenotype, we would see that 9 of them end up being the yellow and the round shape. 3 of them right here end up being yellow but wrinkled. 3 of them end up being round but green. And then this one P right here has both recessive traits. It's green and it's wrinkled. And that's where Mendel found that 9:3:3:1 ratio. And this ratio is important to remember because it can serve as a shortcut for other problems. If you're given a dihybrid cross, instead of making a 4x4 Punnett square and doing 16 different genotypes, you could just remember that 9 of the offspring should have both dominant traits, 3 of the offspring should have 1 dominant and 1 recessive trait, another 3 should have the other recessive trait with the other dominant trait, and then 1 out of the 16 should have both recessive traits. So a very useful ratio to remember. However, use this ratio wisely. A couple warnings. You can only use this ratio if the question is asking for phenotypes from a dihybrid cross. If the problem, however, asks for a specific genotype, you're going to have to make a Punnett square or use the rule of multiplication to solve for that particular genotype. Or if the problem involves parents that are not hybrids, that ratio no longer applies, and so you've got to make a different Punnett square or use the rule of multiplication. Now you might be wondering, what is this rule of multiplication? Well, we will find out about that in class. It's a great shortcut, but for now, just keep in mind the 9:3:3:1 ratio.